In today's video, we're going to talk about biotechnology. An example of biotechnology is this, where we take a green fluorescent protein from a jellyfish that make jellyfish glow green and insert it into an insect to make the insect glow green. So we have DNA from one organism, the jellyfish, being inserted and expressed into a different organism, our insect. Now, this insect has DNA from two different sources. It has its own insect DNA and it has DNA from the jellyfish. This is a special case. This individual is said to be a transgenic organism. Any individual that has DNA from two or more sources is a transgenic organism. Here's another quick vocab for us before we get out. Recombinant DNA. Recombinant DNA is a DNA molecule that comes from two or more sources. But that's just a quick little bit of vocab getting back to our overall big topic of biotechnology. What is biotechnology, though? Well, let's break down the word. Bio and technology. Bio means life. Technology is the application of scientific knowledge for practical purposes. In other words, let's take all the stuff that we learn from science and apply it to our daily lives to make human life a little easier. Some examples of this is farmers have done selective breeding in plants and animals for years, decades, centuries, probably even millennia since the beginning of agriculture and farming to begin with. It's hard to walk in a grocery store and not see some sort of selective breeding or biotechnology in the grocery store, especially in the produce section. Genetically modified food, genetic cloning is biotechnology, as well as bacteria making human insulin. Those are all examples of different types of biotechnology. We're going to take a couple of timeouts as we go along just so we can infuse some vocabulary that we need. But these are the basics, so hopefully we can follow along in our notes. So selective breeding is when humans pick which animals to mate and have offspring based on specific traits. So if we were to take a large bull with a large cow to get large calves, that would be an example of selective breeding, right? Farmers would want to make sure that they have the biggest cows, technically bovine, that they possibly could have because they get paid on price per pound. So they want the biggest cows and bulls that they possibly can get. Another example over here is with corn. Corn has been selectively breeded because, if we have a corn kernel, it looks something like this. The inside is where all the nutrients are, and the outside is indigestible. We've been selectively breeding corn for ever and a year as well because the good nutrients usually have weak outsides, and the ones that have the strong outsides don't have that many nutrients. So they've been crossbreeding these and selectively breeding different types of corn to get a good husk that's going to protect against harmful things with good nutrients. Gene therapy is our next type of biotechnology, and most people refer to these as the knockout genes. Basically, gene therapy replaces bad or harmful alleles and genes with good or beneficial alleles and genes. There's two different ways that we can do this, either with direct or cell-based delivery. In cell-based delivery, what we have is whatever gene that we need is inserted into some sort of vector, which we don't need to know about right now, and we take stem cells from the individual and grow the stem cells in a petri dish with this gene, the good gene that we talked about here. So after the cells have divided and multiplied, they've taken in that gene of interest, and then we reintroduce those cells that were grown in the petri dish with that good gene back into our individual. The other way to do it is the direct delivery, and that's where we take, again, our gene of interest, put it into a vector, in this case a virus, and then inject that gene of interest into the patient. And the whole thing is, if here's the good gene and here's the bad gene, okay, remember that bad gene is part of this big long chromosome. What will happen is the good gene will come in and kick out the bad gene, so the good gene now replaces that bad gene in the chromosome. Unfortunately, there's a pretty low success rate. It's usually only about 10%. Retinoblastoma, I've called before the poster child of gene therapy because it has a astronomically high success rate for gene therapy at 25%. Polymerase chain reaction is our next type of biotechnology, which we abbreviate as PCR, polymerase chain reaction. The biggest thing that we need to take away from this is that it amplifies the DNA. Well, what does that mean by amplify? Well, just like an amplifier makes a lot of more of the same sound. You plug in a guitar to an amplifier, there's a lot more of that same sound. So if it amplifies DNA, it takes and makes a lot more of that same DNA. In essence, it's artificial DNA replication. It's DNA replication that's happening outside of the body. It's helpful in a lot of different things in helping to make sure that there's enough DNA to make a DNA fingerprint, like if there's a criminal case that they found DNA at the crime scene on your favorite show, CSI. You see this all the time. They need to make sure that they have enough DNA so both the prosecution and the defense can run their own DNA fingerprints. So they'll take the DNA and run it through PCR to make sure there's enough copies. Another thing that PCR is useful in is sequencing genes or sequencing DNA so we can know the exact sequence of A's, T's, C's, and G's in a given gene. So all of that stuff happens using a PCR machine. And this picture right here is a thermocycler or a PCR machine. Kind of the process is we have our normal DNA, and remember I just said thermocycler. So we got a whole bunch of cycles, and we're using different temperatures to make all this work. So we're going to relax the DNA and unstring it, so to speak, and break the hydrogen bonds. And then we're going to take chunks of that, and those free-floating nucleotides are going to come in and match it. And basically, all we're doing is making two brand new copies of the same DNA molecule. So it really is artificial DNA replication. Our next piece of biotechnology is gel electrophoresis. Very, very, very important. And the thing that we have to know about gel electrophoresis is this guy right here. Gel electrophoresis separates molecules by size, separates molecules by shape, and separates molecules by charge. Very, very important. If we learn nothing else from this video, make sure that we know that gel electrophoresis separates molecules by size, shape, and charge. It does so by electricity, a sugar gel, and a buffer. So over here, we have our gel electrophoresis machine. We have this box, which is our electrical outlet with our cathodes and anodes running out of it. So over here would be our positive end, and over here would be our negative end. So there's going to be electrical current running through it. Now this piece right here inside, it's kind of hard to see in this one. Okay, this is our gel. And somewhere in here there's things called wells or little holes that we have inside of here so we can put our samples of whatever molecules we're trying to separate in. In our case, we're primarily focusing on DNA. Notice I put DNA at the negative end because DNA is negatively charged. So if DNA is negatively charged, it's going to be attracted to this positive end down here, and therefore it will travel in that direction. Now, the sugar gel, so if we were able to look at it through a microscope and look at the gel, there's all these sugar molecules that are in the gel and make up the gel. and help solidify it. Kind of think of Jello. It's very similar in consistency in what it's like. So we said that we're separating by size and shape. Obviously, we have charge taken care of because we talked about that being electricity going through it. So negatively charged molecules will go to the positive end, positively charged molecules will go to the negative end. So our sugar gel is going to help separate the molecules by size and shape. So here we have this jello-like thing, our gel, with a whole bunch of sugar molecules in it. And I want you to think of a freeway being jammed in a huge traffic jam. Now, I don't know if you've ever done so, but a lot of times little motorcycles are able to bob and weave through the traffic very easily because they're very, very small molecules. So they can get through a traffic jam very quickly and move a pretty good distance in a given amount of time. However, if you took a big semi-truck, a semi-truck would probably bounce off here and bounce off here and bounce off here, and they don't get through the traffic jams very well. They move very, very slowly because they don't want to bounce off anything. They just want to make sure to avoid, so they're going to take their time as they need to get through the traffic jams. So in other words, large molecules move slowly. Small molecules move fast, just like motorcycles move fast and big semi-trucks move slow. The next thing would be in terms of shape. If we have something that is very aerodynamic, like this green guy right here, okay, it's probably going to be able to, again, bob and weave fairly easily. But if you had something that, I don't know, was of the same volume, but was shaped more like this guy in blue might not get through that traffic jam very well because, well, it could get hung up on one of those sugar molecules. So clunkier shaped molecules will move a little bit slower. More aerodynamic molecules will move a little bit quicker. The last thing that we need to talk about is this guy here, is our buffer. So if we were to take and look right here and get eye level with our gel electrophoresis here, we would see, so here's our tray, okay, and there's a stage right here, and then on top of that stage here is where our gel is. Well, as you can see here and here, so here and here, that's where our anodes and our cathodes, the electricity is going to go in. Well, right now we don't have a way for the electricity to go through the gel. If we hook the electricity up straight to the gel, it would fry the gel and that wouldn't work. So what we need to do is we need to add a buffer. And our buffer is a liquid that has a whole bunch of positive and negatively charged ions in it. And what this does is it allows the electricity to flow through. Just like you have a whole bunch of water with positive and negatively charged ions, and they always say, hey, if it's lightning outside, get out of the water. It's because water is going to conduct that electricity because of the positive and negative ions in it. Same thing here with our buffer. We need something to what they call close the system and allow electricity to be able to flow through the gel. Although gel electrophoresis works for dang near any molecule, our primary focus is going to be to use in DNA fingerprinting and DNA sequencing, even though gel electrophoresis is used in a ton of applications. Okay, so here's our first timeout. We need to quickly talk about endonucleases. And unfortunately, this is another concept where you have to know it by two names. Restriction enzymes and endonucleases are the same thing. What their job is to cut DNA at a specific 6 base pair sequence. These guys are naturally made, and there's tons of them. Here's a small list over here. So if we look at this guy right here, BAMH1, BAMH1, its 6 base pair sequence that it recognizes is GGATCC. So it's going to cut here, come down, cut the hydrogen bonds in the middle, and cut here, and leave this left over. See, cut between the G's right there, cut between the G's right here. EcoR1 is another one. Here it is. Its recognition sequence is GAATTC. So it'll cut between the G and the A, come down, cut the hydrogen bonds, and cut between the G and the A again. Now, what I'd also like you to know is that this is a palindrome, meaning it's the same sequence forward as it is backwards. Well, hold on a second. Forward I have GAA, and right here it's CTT. So how is that a palindrome? Well, DNA has a specific direction to it. So if we go in this direction, GAATTC, when we look for the palindrome, we're coming back this direction. So GAATTC, GAATTC. It's the same forward as it is backwards, which is a palindrome. The other thing that I want you to notice here is now we have a segment here and a segment of DNA here. So we have two different pieces of DNA where we used to only have one after our endonuclease cut the DNA. Each piece or cut piece of DNA is called an RFLP. You don't need to know that that stands for restriction fragment length polymorphism. All you have to know is that this piece, a cut piece of DNA, each one of these pieces is its own RFLP. Last thing I want you to pick up here is sticky ends. Down here, you notice that where we cut between the G and A and cut between the G and the A. If we were to have these, if we wanted to put these ends of this RFLP back together, we could, because this A is going to match up to that T, this A matches up to that T, this T with that A, this T with that A. So our base pairing rules are always going to match up these sticky ends together. They want to match up. They have the same exposed base pairs, so they'll want to overlap. That's why they call them sticky ends, so they can overlap and use their base pairing rules to match up. So let's take that information into our next piece. DNA fingerprinting. Now, when most of us think of the word fingerprint, we think this guy right here. And that is not what we're talking about by any stretch of the imagination. We are not talking about a normal fingerprint. The thing about it, and the reason why, is because this fingerprint right here only has to do with a pattern on the skin. While, yes, certain genes and gene expression are responsible for the pattern in this, that's only dealing with one small fraction of all of the DNA. In a real DNA fingerprint that looks like this is a real DNA fingerprint, we're taking care of all of the DNA, all of the genes for all of your body's cells. So that's why we're not talking about that guy, we're just talking about this. The DNA fingerprint, all of the DNA. So here's how DNA fingerprinting works. First thing that we need to do is we need to cut the DNA using our endonucleases. So we're going to cut all of the DNA into, what's that term? Oh yeah, RFLPs. Then we're going to take those RFLPs and put them into a gel electrophoresis. So we're going to load them into the wells on the gel and then put our current or electricity through the gel. Remember, we had that buffer to help close the system. We're going to let it run for, I don't know, maybe 15 minutes or so. And the RFLPs are going to separate in the gel. And it makes these bands. So what could you say about these two bands that you couldn't say about these guys? Well, these guys here would be small molecules, right? If they started up here and they were working towards that end, these ones would be small. Here's our motorcycles. They were able to bob and weave through the gel very quickly. Whereas up here, these would be our large molecules, our large RFLPs, or our semi-trucks. They weren't able to get through the traffic jam. And then maybe these guys in here are somewhere in a medium range. So here's kind of the cartoon version of what our real actual DNA fingerprint would look like. Now that last picture is a little bit difficult to actually tell. So here's a different type of cartoon that represents the same concept. So here we have mom and dad. And here's their DNA that's already been cut and run through gel electrophoresis. And here's those bands, or those markers of their RFLPs, or their chunks of DNA. Here's a simple formula that we got to remember. Okay, mom plus dad equals kid. Another way to put this would be our victim plus the criminal is equal to our crime scene blood. There's a lot of different ways that they like to throw questions at you like this, but if you can remember these two basic principles, you're going to get either question right, whether it's who's the father or who's the mother of this particular child, or who is from these list of suspects, who is the criminal. So here's what I mean, and all it is about matching these RFLPs. So let's just look at this first daughter. Her RFLPs have to come from either mom or dad. Well, as we can see, those match up. So this one came from mom, this one came from dad, this one came from dad, and this one came from mom. So there's no question in any way, shape, or form that daughter number 1 is the child of mom and dad here. Now let's look at daughter number 2. Well, this one maybe might come from dad, but it's a little bit off, so they'd want to do more genetic testing. Same with this one. It's close, but it's off. Some people would say, no, that is not, daughter number 2 is not the child of mom and dad here. Even though this band matches up perfectly, and, even though this band matches up with that one, even though those bands match up, because we have these different ones, daughter number 2 is not the child of mom and dad. Son 1, that came from dad, that came from mom right here, that came from dad, that came from dad, that came from mom right there. So even though mom has this one and the son doesn't have it, that doesn't mean anything. We just need to make sure that the kids' bands match up with mom and dad. Last one is, here's our son number 2, and we have this band right here. Let's just look at that one. This band, if we draw all the way across, whoa, there's nothing there on dad, there's nothing there on mom. This child number 2 is definitely not the son of mom and dad, because this RFLP right here did not come from dad, didn't come from mom. There's no way he could be the child of mom and dad here. And that's how they do DNA fingerprinting, to determine paternity or to determine whether or not a particular suspect did a crime. Last one is probably the coolest one, bacterial transformation. Bacterial transformation, we take a gene from one organism and put the gene inside the bacteria and then have bacteria express the gene. Well, hold on. We took a gene from one organism and put that gene into bacteria. Remember that term? Transgenic organism. Right? From recombinant DNA. So here's another application of that. Bacterial transformation is used to make human insulin, which is a big, huge key one. It's also used to study pharmaceutical drugs and different antibiotic resistances, and also in some chromosomal studies to see if genes are linked. But our big one here is make human insulin. I'm going to show you that now here. Now, before we go into it, again, we need to stop and make sure we know our vocab. We're going to use endonucleases and restriction enzymes again. We're going to use plasmids. Well, if this is a bacteria cell with its plasma membrane and cell wall and capsid, bacteria don't have chromosomes, but they have a nucleoid region. So they have all their, if it was called a chromosome, would be here together in a big, huge group here. Okay, but they also have these things called plasmids. that are small circular pieces of DNA. Here, let me use another color here. So small circular pieces of DNA, those are plasmids, and they can have a bunch of different plasmids on them. It's all just circular, small circular pieces of DNA. Next one is antibiotics. Antibiotics, remember, kill bacteria cells. Antibiotics kill bacteria. And then our last term is a gene of antibiotic resistance. So let's say that this Pac-Man guy right here is an antibiotic, and it's going to go over and take care of this bacteria cell. But this bacteria cell has a gene of antibiotic resistance. So the gene, which codes for a protein, will make the protein and provide some substance that's basically going to clog up the antibiotic, so the antibiotic won't work on the bacteria. If Pac-Man's mouth is closed, or full in this case, it can't eat the bacteria. So gene of antibiotic resistance allows the bacteria to fight against and make the antibiotic completely harmless. So let's take all of that in as we go back to bacterial transformation. The nuts and bolts of bacterial transformation is we're going to take a gene and cut it out with an endonuclease, put it into a plasmid, and then try and get that plasmid into the bacteria cell. And the bacteria cell is going to replicate and make more copies of that particular plasmid. Here's a better picture of it. What we're going to do is we're going to put a gene of antibiotic resistance into the plasmid. So there's the plasmid. Then we're going to take another endonuclease and cut open that bacteria. We're going to use the same endonuclease. It must be the same endonuclease. And we're going to cut out the gene that we want in that plasmid. So we cut out the gene using the same endonuclease for all 1, 2, 3 cuts. And we're going to put that in to our plasmid. Now, how do we put it in? Well, remember that thing about sticky ends where we had GAATTC, C, which means GAATTC. So if we cut here and cut here, remember those sticky ends, they're always going to match up. That's why we use the same endonuclease for all 3 cuts. So when we cut it here, we have our AATTC hanging out on one end and our CTTAA hanging out on the other end. Same thing's happening here on both sides. So because of these sticky ends and using the same endonuclease, because of those base pairs are the exact same, they'll match up perfectly when they go to combine. Now we have our plasmid with our gene of antibiotic resistance right here, the gene that we want, like the human insulin gene, and now we got to get this plasmid into the bacteria. Remember, they have 3 layers of protection, bacteria cells do. So they can't just come in and knock on the door and say, hey, can I come in? They need to have a special process called heat shock to get the plasmid inside the cell. So what heat shock is the first thing that we do is we put everything on ice. We get it really, really cold. And when things are cold, they come together really, really quickly. We're going to leave it in cold for about 10 minutes or so. The next thing we do, we put it in heat in a water bath. So what that causes is cold, 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 everything comes together, and then a really quick expansion into heat. And like I just mentioned, when you put it in heat, things expand. And if you do this really quickly, the chances of a hole happening in these layers is pretty good. So the plasmid will sneak in those holes in that particular time when we went cold and then hot really quick. So the plasmid will sneak into the bacteria, and if the plasmid is inside here, we want it to stay inside. So we're going to put those cells back on ice and cold. So we go cold, 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 bring everything together, then hot really quick, and hopefully a hole opens up for the plasmid to sneak in. We want the plasmids to stay there, so cold again. And then last thing that we have here is, so now that this plasmid is inside of the bacteria cell, this bacteria cell doesn't really know any better. So it's going to replicate and divide, and everything's wonderful. We can express this gene. If it's human insulin, we can make sure that the gene is expressed, so the bacteria cell now makes human insulin because it has the human insulin gene. Now, one last thing here, and that is, remember, when we talked about this heat shock process, we said hopefully a hole develops for this plasmid to go in. So when we're growing these bacteria cells that hopefully have the plasmid, not all of the bacteria cells are going to have the plasmid. So here, down here is our food in our petri dish that we're growing our bacteria on. Now, also remember that we said we had our gene for antibiotic resistance. We also, in addition to food, we also put in an antibiotic into the food. So genes like here, or bacteria cells that do not have the plasmid, the antibiotic will kill off, right? Because we don't want to grow bacteria that didn't get the plasmid. We only want to grow bacteria that did get the plasmid. But also remember our gene for antibiotic resistance in our bacteria that did get the plasmid here in blue. They have a gene of antibiotic resistance, so the antibiotic won't do anything to those bacteria. The bacteria that took in the plasmid have the gene of antibiotic resistance, so will therefore grow. This is obviously a phenomenal piece of biotechnology. Hopefully we can remember all the different types of biotechnology that were covered today. And that is all she wrote.
It's like this and like that and like this and it's like that and like this and like that and it's like this. So just chill till the next episode.